Hi, in today's video, we will be learning more about biomaterials by taking the example of a very popular implant called the intraocular lens. So what is the intraocular lens and where is it used? The intraocular lens is a small artificial implant that replaces the eye's natural lens, which is removed during cataract surgery. Now you might have heard of cataracts. It's a vision-hindering condition that occurs due to the natural lens inside the eye becoming cloudy. Fortunately, cataracts can be treated with a surgery during which the defective lens is removed and replaced with an IOL. Lens epithelial cells, also known as LECs, are responsible for the growth and development of this natural lens. So let's find out what are the different kinds of materials used for intraocular lenses. Modern-day IOLs are usually made up of acrylic and silicone materials. But early forms of IOL were made up of PMMA, which was a very rigid material requiring surgeons to make large incisions to place it in the eye's capsule. Therefore, in recent times, foldable IOLs manufactured from acrylic materials have become increasingly common. Alongside flexibility, IOLs are also characterized by their hydrophobicity or hydrophilicity, refractive index, and surface charge. How do cells adhere to materials and what components of the cell structure support this adhesion and subsequent behavior? All cells and tissues are surrounded by a scaffold of non-cellular components called the extracellular matrix. It is an extensive network of different proteins and other macromolecules. The ECM composition of the lens capsule mainly includes structural proteins such as collagen 4, laminin, nidogene, and perlequin. Cells can bind to these molecules in the ECM through specific junctions on their surface, which provides them with mechanical support and anchorage. These junctions are made up of transmembrane proteins called adhesion receptors. An example of an adhesion receptor is integrin. Our cell also has a skeleton that helps hold its shape, much like our bodies. This is made up of specialized protein filaments that extend throughout the cell, known as the cell cytoskeleton. The cytoskeleton is made up of three main filaments: actin filaments, microtubules, and intermediate filaments. Actin filaments are mainly found near the plasma membrane. The point where the cytoskeleton connects to the plasma membrane is called a focal adhesion. Integrin anchors actin filaments at focal adhesions to the ECM proteins, and hence allows the cell to attach to the proteins adsorbed onto the biomaterial. What happens after the IOL is implanted into the eye capsule? The process of IOL implantation leads to disruption of the blood-aqueous barrier. As a result, proteins in the ECM and from the blood plasma are adsorbed onto the IOL. These mainly include proteins such as fibronectin, collagen type 4, collagen type 1, alongside other proteins. Following this, LECs migrate onto the lens capsule. The adsorption of these molecules then allows for LECs to adhere onto the IOL. LECs consist of cell adhesion receptors such as beta-1 integrin, ICAM1, and CD44. These allow LECs to attach and grow onto the collagen and fibronectin. Other cells may also attach to the IOL, such as fibroblasts via collagen 1 and macrophages via vitronectin. Let's understand how cells communicate with each other following the implantation of a material. Cells can communicate with each other by releasing various signaling molecules such as hormones, neurotransmitters, or local transmitters. These may be in the form of proteins, peptides, amino acids, or other small molecules. Cytokines are a type of cell signaling molecule which are mainly characterized as local transmitters and are involved in signaling between cells during an inflammatory and immune reaction, such as one that occurs after the implantation of an IOL. Each cell has many signaling receptors on its surface which bind to specific signaling molecules. The signaling receptors are activated once they come in contact with the cytokine and further stimulate other proteins that lead to a specific cellular response, such as stimulating the cell to produce inflammatory mediators or migrating to the site of damage. The primary cytokines involved in response to IOLs include interleukins, transforming growth factors, fibroblast growth factors, and prostaglandins. Communication between cells can occur either with cells in direct physical contact or other nearby cells, or even with self. Cell-to-cell communication between cells in nearby area is called paracrine signaling. For example, immune cells release cytokines such as interleukin-1 and TNF-alpha, which are produced in response to tissue damage. These may bind to other immune cells and recruit them to the local implantation site. Cell-to-cell communication between cells in direct physical contact is called contact-dependent signaling. This happens through integrins on the surface of LECs that are attached to adjacent cells. Cell-to-cell communication also happens amongst LECs when they release signaling molecules which are then bound to receptors on their own cells. This includes TGF-beta, which is released in response to inflammation. This influences cell differentiation and proliferation of LECs and fibrosis. How can biomaterials be tested prior to implantation? Many tests can be employed to assess the biocompatibility of IOLs. These mainly include cytotoxicity and irritation tests. These can either be performed in vitro, which means in a lab petri dish, or in vivo, which means inside a living organism, such as an animal model. Irritation tests focus on immediate inflammatory responses at the site of contact with tissues, while cytotoxicity test assesses the broader cellular responses and viability after exposure to materials. IOLs are mainly tested in accordance with EN ISO 119795, which highlights 4 main tests for assessing the biocompatibility of IOLs: cytotoxicity, irritation, genotoxicity, and local effects after implantation. Assessing the cytotoxicity of IOLs includes 2 primary tests: direct contact test, which involves placing the IOL material directly in contact with the cultured mammalian cells. Second is elution test, which involves exposing the IOL to an aqueous environment, then testing the reaction of cultured cells to the extract. The direct contact test may be performed using a method called MTT assay. This involves use of MTT solution, which can be adsorbed and reduced to a purple insoluble product only by metabolically active cells. The formation of this purple product is hence directly proportional to the number of viable cells. For the procedure, human lens epithelial cells are harvested and cultured in appropriate media under controlled conditions. The cells are then seeded onto well plates where the implant material, example PMMA, is deposited. MTT solution is added to the samples and the cells are incubated for an appropriate period. The amount of purple product formed can be assessed using a spectrometer that reads the absorbance of light through the samples. Elution test involves immersing the IOL in a suitable solvent such as a saline solution for a specific duration of about 24 to 72 hours at an appropriate temperature of about 37 degrees Celsius. This allows any leachables or soluble components from the IOL to diffuse into the solvent. This setup stimulates long-term exposure of many years of the implant to aqueous material. Any product particles can be tested using chromatography. In order to test the effects of extracts in vitro, the extract can be diluted and applied to cultured cells. Following this, the cell viability can be assessed using methods like MTT assay. This approach stimulates how substances might leach from the IOL into surrounding tissue after implantation, providing critical information about potential long-term effects. Let's discuss irritation test. This is performed for intraocular lenses to evaluate the potential to cause eye irritation upon implantation. In vivo testing involves IOL being administered into one eye of a healthy albino rabbit. The eyes are then observed for signs of irritation up to 72 hours. Genotoxicity test is done for IOLs to test their genotoxicity, carcinogenicity, and reproductive toxicity. The steps typically involved preparing an IOL extract by exposing the IOL to appropriate solvent over a specified temperature and period, similar to an elution test. Then, a series of in vitro tests are performed, including bacterial reverse mutation test, chromosomal aberration test, and mouse lymphoma assay. Local effects of implantation can be tested using rabbit models. In one study, healthy New Zealand white rabbits were used. PMMA was implanted into the capsular bag of the rabbit's eye after performing a cataract surgery. Some eyes were also subjected to sham surgery without eye implantation to serve as a control group. Clinical observations were made to assess for any signs of inflammation, opacity, or other adverse reactions. Then after about a period of 6 to 12 months, the rabbits would undergo histopathological evaluation to assess the tissue for cellular responses such as inflammation or foreign body reaction. What are the adverse effects that have been reported following the implantation of a biomaterial, and how can they be avoided? Posterior capsule opacification is the most common complication following the cataract surgery with intraocular lens. It occurs when the posterior capsule, which is the thin membrane that holds the IOL in place, becomes cloudy. This happens when LECs migrate into the posterior lens capsule and differentiate into fibroblastic and lens fiber-like cells and form a cloudy layer of tissue behind the IOL. The use of PMMA in IOL greatly increases the incidence of PCO because it is a hydrophilic material. This means it has considerably lower affinity to fibronectin and hence develops a weak attachment to the capsule. This allows for a gap between the posterior capsule and IOL where the lens epithelial cells can migrate and proliferate more actively. On the other hand, hydrophobic acrylic lenses only develop PCO 21% of the time compared to 74% for PMMA. The hydrophobicity of hydrophobic acrylic lenses allows for much higher affinity to fibronectin. The adsorption of fibronectin onto IOL results in a strong attachment of the IOL to the capsular bag. This forms a sandwich-like structure between the capsule and the IOL surface. The formation stabilizes and prevents further proliferation of epithelial cells behind the IOL, resulting in lower rates of PCO development. How does the immune system contribute to the body's response to a material, and how do materials interact with the blood? Upon implantation, surgical procedure disrupts the blood-aqueous barrier, leading to an influx of proteins and immune cells into the anterior chamber of the eye. The immune cells attached to the proteins adsorb onto the IOL, recognizing it as a damage or non-self, which gives rise to inflammatory response. Part of the innate immune system includes a series of proteins which can react in sequence to produce an immune response from the cells. This is called the complement, a non-specific recognition and elimination of foreign body elements from the body. The complement cascade can be activated by specific proteins called complexes. Examples include C1 complex, C3 complex, and mannose-binding lectin. These protein complexes are present in the blood. There are three types of complement activation pathways: classical pathway initiated by C1 complex, lectin pathway initiated by MBL, and alternative pathway initiated by C3 complex. It has been shown that different materials might trigger the complement through different pathways based on their affinity to specific complexes. For example, PMMA has been shown to have lower affinity to C3A, while silicon has been shown to have higher affinity to C3A. Hence, silicon has been shown to induce a complement activation by the alternative pathway. The resulting activation of complement system in the case of IOLs may recruit leukocytes such as monocytes to the site. These cells then attempt to destroy the material, which gives rise to the foreign body response. The kind of reaction produced depends upon the size of the material. Since IOLs are about 11 to 13 millimeters in diameter, they are attacked by a fused giant cell deposition. This is to ensure that they can cover as much as possible of the foreign surface. This is followed by the release of extracellular enzymes that attempt to digest the material. The immune reaction can also be triggered by molecules in the biomaterial called pathogen-associated molecular patterns and damage-associated molecular patterns. These can take in diverse forms, for example, polysaccharides, peptides, glycopeptides, and lipopeptides. The surface of biomaterials may exhibit physiochemical features that resemble these molecules. The macrophages recognize PAMPs via receptors known as pathogen recognition receptors. Upon recognition, they may produce cytokines that will further recruit lymphocytes such as T and B cells, which gives rise to the adaptive immune response. It is important to understand that the extent of immune reaction to IOLs is also influenced by its material properties. For instance, hydrophobic IOLs tend to attract more inflammatory giant cells compared to hydrophilic ones. This difference is attributed to the surface properties and chemical structure of the material. The hydrophile and lipophile balance and hydrophobic parts of the material can also vary the stimulation of innate immune cells through PRR, which causes the inflammatory response. I hope this video gives you a good idea of how biological systems interact with biomaterials, such as the intraocular lens, and how we can enhance their properties to suit our desired function. Thank you!